The purpose of this talk is to walk you through the process of choosing and acquiring the sequences that you're going to use to make a phylogenetic tree. That's the necessary first step in making any tree, is you have to choose the sequences. Not only is it the first step, it is the most difficult part of making a phylogenetic tree. And it's the most time-consuming part of making a phylogenetic tree. And the reason it's most difficult is because in order to choose the sequences, you have to know why you're making that tree. Here are some bad reasons to make a phylogenetic tree. Because my advisor told me to. That's probably a bad reason for almost anything. Because a reviewer insisted on a tree. Because all the papers in the intended journal insisted on a tree. These reasons really treat a phylogenetic tree as though it is some sort of decoration for a paper, or because such that the tree is the end in itself. A phylogenetic tree is not the object of a study. It's a tool. It's a tool to understand a biological process, and you have to make that tool. And it makes no sense to me to try to make a tool if you don't know why you're making it. So, for today's example, the goal is going to be to ultimately make a tree of the homologs of the EBGC gene of E. coli. And the first question is, why do you want a tree of the EBGC genes? If I can't answer that, I should stop and go home. So, for my purposes, I want to make this tree to eventually estimate the ancestral sequences, the sequences of ancestral EBG proteins. And that brings up another question. How distantly related should these sequences be? How exhaustive do I want to make this? For my purposes, I want to get sequences that are as distantly related as possible. How representative should they be? Do I want to put onto my tree every known homolog of the EBGC gene of E. coli? Well, I do not, because that will be a very, very, very large tree. I want to get a tree that is very representative of the homologs that are out there. So I choose a criterion as no more than 4 species from each genus, but I want as many genera as possible. How do I ensure that all of the sequences are homologous? This is critical, because the basic assumption of a phylogenetic tree is that every sequence is on in there is descended from a common ancestor, which is what we mean when we use the word homology. Not similar, but homologous. The best tool to ensure homology of the sequences that you choose is to use BLAST from NCBI. And BLAST will produce for you a list of putative homologs of your query. And from that list, how do you identify and choose the sequences that you want? And the answer to that is manually. You actually have to look at the list. The starting point for a BLAST search is a gene sequence. In this case, it's going to be the sequence of the EBGC gene of E. coli K12 strain MG1655, for which we have an accession number. And I guess I have a pointer here somewhere. Yeah. So I've got an accession number. That's the accession number for this genome. But I don't want the whole genome as my query. I want just that part of it that is the EBGC gene. That is these bases. So we're going to open Start Mega. And when you start Mega, if you go under the Align menu, the first thing at the very bottom, you will find Do BLAST Search. This is the opening main Mega page. You choose Do Blast Search, and Mega opens its own browser and immediately takes you to the BLAST page, the BLAST analysis page at NCBI. That page allows you to do a number of different kinds of BLAST searches, and the default in Mega is this tab that says BLAST N. There are three important parts to this page where you set up the conditions for your BLAST search. The top part is where you specify a query sequence. This is the sequence that BLAST is going to try to match to find homologs. I will in here put in this text box the accession number for the genome, and over here the range of bases. And then it gives me a job title. I could just as easily have pasted the coding sequence for EBGC in here, but then it wouldn't do this cute business of giving me a job title. So once I've got that, I've got a query sequence. Now I have to tell NCBI and BLAST where to look for those matches. That is, what database to query. There are a lot of choices. You can pick human genome, mouse genome, or for those who are not interested in those uninteresting organisms, others. Go down here and click, and you get a bunch of choices, and one of them is the nucleotide collection, the non-redundant nucleotide database. So we'll select that, and just to simplify things, we'll exclude models and uncultured samples from the list. And so now we know where to look, we know what to look with. And the last thing we have to choose is the program selection. Do we want to look only for sequences that are highly similar to our query? No, I wanted to look as widely as possible. We could look for more dissimilar sequences or somewhat similar sequences. I'm going to choose somewhat similar sequences. So these are the major parameters to set up, but I've got one more thing I want to do. I want to go down here and click this thing that says Algorithm Parameters. And that takes you further down in the page, or opens that part of the page. Here is the max target sequences. That's how many matches do you want me to find in that database? And the default is 100, which was perfectly good 20 years ago when the database had about 6 sequences in it. But now with millions of sequences, that's completely inadequate. If we're searching with a query that's from E. coli, there's a lot more than 100 sequenced E. coli genomes, so everything you get is going to be a perfect match and be useless to you. I routinely change that to 5,000, just to make sure that I will search far enough down to get distantly related sequences. And when that's done, go down here and click the BLAST button. Do not tick that thing that says Show Results in a New Window, because we're in the Mega browser, and doing that will take you places you do not want to go. It will show you the results in a window and you'll be happy. Just click the button. After a few minutes, the BLAST result page is returned. It tells you what the query ID was. It was a DNA molecule and what the query length is. And that's a very nice thing to know. And down here, it tells you how many hits, that is, sequences that match your query, it found. 992. 100 would not have been enough. And then there's this graph, which I'm not going to talk all about because it's essentially useless in these circumstances. Immediately below that graph is what we have to work with. This is the list of the hits that it's found. And for each hit, there are a bunch of things. There's a column that contains a description. The description here is Escherichia coli VH1 chromosome complete genome. That's also a link that we'll use later. Then there is max score and total score. Neither of these are terribly useful to you because the absolute values of those scores depend upon the length of the query as well as how well it matches. But query coverage is very important. How much of the query matched the region that matches in this chromosome? And the answer is 100%. That's good. I've got something that aligns over the whole length of the query. And then there is the e-value. The e-value is the likelihood that you will find, it is the number of matches that you are likely to find that match by chance alone, given a query of this length and a database of that size. That's not exactly the same thing as the probability that this sequence is not a homolog, but you can certainly think of it that way and you won't be very far off. So here, what this is telling us is this match has no probability of not being a homolog, and what's more, its identity is 100%. This is exactly the same as the query. So we want to add that to say, we're going to put that on the tree. Question now becomes, we found something we want, how do we get it? Go back and click the link that is the description, and it will take you down below the list to the alignment. And here's the alignment. And that's the query sequence. This is the subject. That means the hit. And they align perfectly over the entire lengths. The 450 base pair identity, 100% identical. The e-value is the same as what you saw before. There are no gaps. It's perfect. How do we get it? You go up to this word GenBank here that's circled. It's a link. Click that link, and in the browser, it opens a new page on a new tab. So now, here we were looking at the results page. This tab that we've got visible now is showing us the GenBank file for that genome sequence, and it says this part, this range of bases, is the match. Okay. Let's go down and see if we think we've really got a real match. Look at the bottom. And the question here is this DNA sequence that it claims to be a match a coding sequence? We are searching with a coding sequence. We want to put coding sequences in so that we can make a tree from those coding sequences. Well, it starts with ATG and it ends with TAA. It's a coding sequence. So now we really know that we want it. Go back up to the top. You could have done every bit of this without Mega. You could have done this through any browser that you want, and in which case you would have had to click that little thing, open it up, choose Save File. It would have saved a file, given it a name, something like sequence, just a generic name that has nothing to do with anything. Then you would have had to open that file, copy the alignment, copy the sequence out of it in FASTA format, paste it into another file where you're going to start accumulating different sequences. All of that's a pain. Mega, the reason to use Mega for this part of the process is this magic button right here. If you click the button Add to Alignment, it takes care of the whole thing for you. When you click that button, it opens a little dialog box, and the only really important thing to you is right here. Sequence label, manually editable. And the name that it automatically gives for this sequence is Escherichia coli VH1 chromosome complete sequence. That's what's going to appear on the tree. And that's a very long label to label something with a tip node on a tree. So you want to edit that so it's much shorter. We're going to edit it so it reads Escherichia _coli. Underscore, not space, because many downstream applications will throw up and crash if there is a space in a word. So having edited it properly, click OK, and it opens Mega's Alignment Explorer. And you see there is the sequence, Escherichia coli, and you see the beginning of the sequence here, ATG, We have now completed identifying the sequence that we want and acquiring that sequence. And we only have 958 to go to look at it. So, should know the rules for naming sequences and stick to these rules. Each name must be unique. Every time you add a sequence, you have to give it a name that is different from that of every other sequence you have added, because otherwise your tree will have more than one sequence with the same name, and most things will throw up at that point and tell you that this is an error. No spaces allowed in names. I mentioned that already. Indeed, you should only use letters, numbers, underscores, and the dash character in names, because you may want to take the results of this and put it into another application, some command line application that is going to be impossible to look at anything else and be confused by it. And the final thing is, make the name something that is meaningful. Don't give your sequences names like John and Mary, and don't even just give them names that may just be something that your lab uses, a slang term. Give them names that are appropriate to the data set and that will be meaningful to you and other people. So now we're done with that. We've added it. We'll go back to the file, and here's that tab that we're looking at. We're just looking at that file. And the first thing to do is close that tab, which you can do by command selecting it and control clicking, or just hitting Control W. It will close that tab and take us back right where we were in the output file. So that's what we've added. If you go back and click this word descriptions, now it will take you back up to where you left off in the list, which is not so important at this point, but further down in the list, that gets to be a really convenient thing. So here's the list again. We've added that, and now we have to look at every one of these other ones and decide whether to add it. But I have some rules for adding things. And my first rule was I only want no more than 4 species from each genus, and I only want 1 sequence from any species. I've got my E. coli. So I certainly don't want any of these, especially since they're all the same sequence. They match at 100% match. So I will just scan down this list until I come to something that is not E. coli, which, while it technically means I have to look at every item in the list, I don't have to look very long if it says Escherichia coli. In order to decide what sequence I'm going to add, in effect, I have a little algorithm for this. I look at a line and I ask, is that species already added? If the answer is yes, I go on to the next line. If it's no, then I ask, have 4 species of that genus already been added? If the answer is yes, I'm done with that genus and I go on to the next sequence. If the answer is no, then I have to ask, is the query coverage, remember that line there? Is the query coverage less than 85%? For my purposes, I've chosen to set a lower limit of 85%. That's a decision that you have to make based on the reasons that you are making this tree. So that's not a rule, that is a conscious criterion that you have to choose in advance. If the answer is yes, it's got less coverage than that, then I skip that and go on to the next sequence. The final one is the E value less or greater than 10 to the minus 4? This is the criterion for homology. We're only going to add things that we are confident are homologs. And in effect, this is saying, if I think that there's more than 1 chance out of 10,000 that this is not a homolog, I'm going to skip it. So if the answer is yes, the value is greater than 10 to the minus 4, because the higher the value, the greater the chance that it's not a homolog, then I'm going to go on to the next sequence. If the answer to this question is no, and I've gotten through all of these, then I'm going to add the sequence using exactly the same operations that I just finished showing you. So here I've gone down through a whole lot of E. coli, and I've gotten down here to where I see Shigella boydii. And I look over here, and the query coverage is 55%. Not interested. Go down to the next one. Citrobacter cosseri, 100% query coverage. 4 times 10 to the minus 126th. That's good enough confidence that that's real. And it's only got 84% sequence identity. It's different from what I already have. That's good. Yes, I want that strain. So I go through the same things. I click the thing, I go down, click the GenBank, bring up the file, look at the bottom of the file, and what I see at the bottom of that tab is TTA, TTGTA, and it ends with CAT. That does not look like a coding sequence to me. And if you look at it, you say, well, that's the reverse complement of the coding sequence. At least it looks like the reverse complement of a coding sequence. You do not want to add reverse complements of your coding sequences to your list, because when it tries to align those, it's not going to find any commonality between a coding sequence and its reverse complement, and that will screw up your alignment to a fare thee well. So going back up to the top of this file, there's a little thing over here that says Customize View. It may be open like that and show you this part of the panel or not, but if it's not open, click that, and you look down at the bottom and there's a line that says Show Reverse Complement. And the first thing you think is, but it's already showing me the reverse complement. But if you tick this, it will show you the reverse complement of the reverse complement, and that'll be what you want. So tick that, and then click Update View. Don't forget to click Update View. And now look down there, and sure enough, we've got a coding sequence again. And we can go up and click the magic button and add that to the tree. Continuing on down, I start to see that this sequence I probably want. It meets all my criteria, 88% sequence coverage. Here's a Klebsiella strain below my cutoff. This Enterobacter one is just above the cutoff, so I'll take that, I won't take that, I'll take this, I won't take that, or indeed any of the rest of these that I see so far. So you do have to look at each of these lines, but it doesn't take long, because you just look, is it something that you want? Is the coverage good enough? Is the E value low enough? And there were 992 hits. 449 of those hits met all of my criteria for being acceptable homologs of EBGC. That is, the E value was low enough, the query coverage was high enough, but I only wanted one sequence from any species and no more than 4 species from any genus. And when I got through, I had a bunch of sequences, but they fell into only 7 genera. All of those were in the family Enterobacteriaceae within the order Enterobacteriales. Now that might be the limit of where the gene EBGC is distributed. But maybe not. Maybe I can find some that are more distantly related. But that's the search. That's all I got. How can I find more distantly related homologs? The answer is to search for homologous proteins instead of searching for homologous DNA sequences, because DNA only has 4 character states, and by the time you get down to approaching 25% sequence identity, you're looking at what is basically a random sequence in terms of how related it is to your query. Proteins have 20 amino acids. That's 20 character states, and you can get down to so you start approaching 5% sequence identity before you get into that random noise. So we're going to go right back to the beginning of this and bring up the search page, accept we're going to click on this BLAST P, so we're doing a BLAST protein search. We're going to search protein database with protein sequences. This is the accession number for the EBGC protein. That gets put there. It fills that in very acutely. I choose as a database the non-redundant protein sequence database. Again, there's here in the pull-down menu, you can choose that. There are a number of different protein searching programs. I'm going to choose BLAST-P, which means I'm searching the protein database with a protein. And I'll go ahead and I'll set the number of hits back up to 5,000. And the results page looks like this. I don't remember how many total hits there were. And it looks very similar to that from the nucleotide, from the BLAST end page. There's a description, scores, query coverage, an e-value, and the percent identity. And this all looks very good. 100% query coverage, a nice low e-value, 100% identical, and the sequence is called multi-species. You look at that and you say, I don't know what to do with that. What is a multi-species? And the way you find that out is you click on the link to go down to the part of the alignment that shows you an alignment of multi-species. And here it is. Here's your alignment. It's an alignment of a protein with the query with this multi-species sequence, and it's identical to it. But there's this line up here that you've never seen before. C5499 more titles. This is a perfect match. What this is telling us is there are actually 5,499 protein sequences out there that are identical to EBGC. Well, that's not so good. What do you do about it? Click that, and it opens a list of all of these. And now we have links to each of the individual 5,499 protein sequences that are identical to that. The first one in the list happens to be the EBGC, the evolved beta-degalactosidase beta subunit from E. coli K12 substrain MC1655. That's a query. So we want to add that. To get to it, click the sequence ID. And that will bring up, just as it did before, the GenBank page for that protein sequence. And there it is. We could go and click Add to Alignment, but we don't want to put the protein sequences into our alignment. We want the DNA coding sequences for reasons that have to do with what you've got to do when you are going to reconstruct ancestral sequences and process those files. So we've got a protein sequence. We want the corresponding coding sequence. If you scroll further down, you see this word CDS, standing for coding sequence, that I've circled. That's key. And it's blue. It's a link. If you don't see that that's a link, stop and go back because you can't use this. Here is the sequence itself. You click that coding sequence, and it highlights the protein sequence, brings up this details thing, but the most important thing it does is it adds this bar down here where it has the word GenBank, which I've circled. That is the link to what you want to get. And it's a very strange and absurd thing for NCBI to have done. And it took a long time for me to figure out, how do I get this? That's it. Click coding sequence, look at the bottom, find the word GenBank, and click on it. And then that brings up the sequence for, that's what we had before. MG1655, different strain, the coding region, and that's wonderful. We're going to scroll to the bottom of the page and see if the right sequence is in the right orientation, not the reverse complement. And if it is, we'll add it. And if it's not, will do the reverse complement. So once you've got to that page, you're going to proceed just the way I showed you before. Here's some things not to do when you are trying to download coding sequences and you're using protein query. You do not want in your list to go over here and click that link where it says accession number. It will take you to the file for the protein sequence, but you won't have a link to the coding sequence, and so you can't get it. So that's a bad thing to click. Another thing that you don't want to do is go down to the bottom and click this link, sequence ID. Because if you do that, you cannot get to the coding sequence. You'll notice here, this wasn't a multi-sequence. It didn't say click here for 5,499 more sequences. Well, that's too bad, because from this one, we can't get where we want to go. If you don't see something that says see n more titles, even if it's just one, you're stuck. You can't get the coding sequence, and so you'll just skip that and go down to the next one. See one more title. Click the sequence ID, and now it will take you to the GenBank page for the coding sequence, and you can add it as you did before. Eventually, after adding all of the sequences that you want to add, you're going to get to the point where you have got all of the ones that you want, and your Explorer looks like this. Got all these nice sequences in there. They're not aligned, they're just there. And the first thing you want to do is you want to save session. You have just spent some part of your life, probably a distressingly large part, making a lot of decisions about whether you want this or don't. You do not want to lose the fruits of all that labor. So save that. You can compare the result of searching with a protein sequence with searching with a DNA sequence. When I searched the nucleotide database, all of the hits that I got were in the gram-negative kingdom of bacteria in the order of Enterobacteriales. That's as widely distributed as those were. When I searched the protein database, I wound up with 40 hits that I took. 36 of those were in the gram-negative kingdom, but they were in 3 different orders, and 4 of them were very distantly related in the gram-positive kingdom. So by searching with a protein query, I was able to dramatically expand the range of hits that I got. And that was well worth doing. Again, the way you handle things and what you choose to do depends on the purpose for which you are making this tree. So, if you're going to make a tree, know exactly why you're estimating that tree. And you have to decide the criteria for choosing what is an acceptable sequence from the blast search that you do. That's your decision. And the application of that decision, of those criteria, is what's going to determine what winds up on your tree. And to detect the most distant homologs of your query, do a protein query with BLAST-P instead of a DNA query. And then apply your criteria for choosing them quite rigorously. It's good to write those criteria down and have them in front of you as you're looking down here, because if there's one thing you want to do, it's you want to be consistent in your choices. When you add a sequence to the tree, before you add a sequence to the tree, before you add it to the Alignment Explorer, be sure to check that it is in the right orientation, that you're not adding the reverse complement of what you want, or you will follow things up to a fare thee well.